Alright. Right. Okay, then we start the today's proceedings. Okay, right. Right. Okay. Right. Then today we are going to start organelle parts. Right. Okay. Organelle parts. So then before the entry for the main organelles, right? Main cellular organelles. First, we have to go through the kind of a rough background and a kind of an entry there, right? So some basic and actually these are some kind of additional facts which are not there in the resource book, but they are very important in explaining the concepts, right? They are very important in explain the concepts. So therefore, you have to just have a proper knowledge regarding those kind of things, right? Very basic facts, but for the proper understanding of the rest of the theory parts, you need these theory points. Otherwise, it will be tough. And normally, these are not taught in normal classes and also not given in the resource book as well, but we are giving it. Actually, these are kind of things we are just using for the revision students with our classes from with the experience of teaching revision students. Actually they lack this knowledge, right? They lack this kind of knowledge because they haven't been going through those kind of things. Okay, they just in majority it means they are actually well prepared students for the syllabus. No, actually they have gone for several classes and maybe tried and went through many classes. So once they are taught with these kind of facts, they get kind of amazed, right? Okay, they say, wow, the first time we are learning this, and these are very smart facts. Likewise, okay, there are many positive feedbacks from their side, right? Okay, anyway, right. Then okay, we'll start from the today's proceeding. So as we normally do, what we are doing, we are uh, writing the topics with the kind of a number, point number. You need to point number this one likewise. But here today's ones, today points are not included in our point collection. These are kind of additional facts. Therefore, only the topic is needed. So therefore, no need to write the topic numbers, right? Okay, we'll start the today's proceeding then, right? Okay. And another important thing as well as now from the beginning we have had some short sessions, and also this month also here with the 2026 batch, those your elder brothers and sisters, we are preparing them for the exam. Okay, so this month is actually really busy with them, right? In a tight schedule. You might have seen the timetable that we have shared in our Telegram official channels of revision official channels, 30-day plan. That is very hectic plan even from our side, from their side. But anyway, we are actually giving our best, very best, in order for this success. Okay. So but after they are after we said them in the exam, actually we are allocating more time towards your class. Actually what we have done, actually from now for these 3 to 4 months, we have done some short kind of sessions, not that much prolonged session. But the most important thing is the session, if it is 1 hour, if it is 1.5 hour, if it is 2 hour, the entire session is about learning and teaching, right? No any other facts like any motivation, any additional facts, any funny things, any jokes, no any silly things, right? Okay, so therefore imagine a normal 3 to 4 hour normal theory class also. Actually if you take the lesson part in the teaching process, there will only be 1.5 hour, um, right? Okay, maybe sometimes 1 hour, because majority of the time there are some additional talking, especially in your theory classes at the early days, right? Okay, this is what you are normally going through. But we are not like that. But after the 2026 batch, they are set up to the exam. Then what we are doing, we are allocating more time, right? By taking that more time also, then doing some extra classes means some prolonged classes, which is around 5 hour, 4 to 5 hour classes, and we are covering the syllabus very quickly, right? Because you are getting your term test. I think by another 3 odd months, you will have your first term test. So prior to that, it means before 1 month for the term test, we are in the final month, just last month of the before your term test, we are actually going through term test revise, right? Right, okay, that final month we are going through term test revise. Before that, we have to complete the syllabus required for the first term a month earlier. That's the plan. So therefore, and also in recent time, we will have A grand seminar, right? You will have a grand seminar. uh, so then you can share it with your friends, right? Okay, and all the other parties of the seminar. Okay, so that is the prospect, or that's the the plan of the class in the upcoming months. Okay, so get ready, uh, right, for prolonged classes, right? Not like short bursts like these classes, right? We are now conducted, right? So these will be somewhat long classes, 4 to 5 hour seminars like things, but they will be interesting, complete interviews, right? Okay, we'll see what will happen then, right? Okay, then for the today's proceedings, we have to start. So first of all, cellular components, right? Cellular components means actually these are the components which are found inside the cells, right? Right. These are not found out. It means no. These are not extracellular components. These are intracellular components. It means inside the cell. Right. Those cellular components are inside the cell. So we can divide those cellular components into two. Right. We can divide these cellular components into two. One is organelles. One is organelles. Right. Organelles. Right. And the rest of the one other things are right. Okay, right? Subcellular components, subcellular components. Now, what is the difference between these organelles and subcellular components? All are found within the cells. Within the cell, the difference is organelles are membrane bounded, right? Okay, membrane bounded. Further, we can divide organelles into two, like as a single membrane bounded. It means organelles which are covered by one membrane, single membrane bounded, single membrane bounded, and the other one is double membrane bounded, double membrane bounded, the other one, right? And subcellular components are actually, these are non-membrane bounded, non-membrane non-membrane bounded, means no membrane covering there, right? Non-membrane bounded, no membrane covering here, non-membrane bounded. Okay, got it, right? So cellular components, actually these are found within the cells, right? These are found within the cells, within the cell, right? Not exterior to the cell, within the cell. Okay, got it, complete it quickly. Hurry up. Right. Okay. Then we'll go through the other part. Now we need to actually figure out the different type of organelles. It means we have to categorize them, right? So what are single membrane bounded? We need examples. And what are double membrane bounded? And we need examples. And further, we need the examples for subcellular components out of the organelles and subcellular components, so the cellular components found within both animal and plant cells, right? Kind of analyzing them, kind of detailing, but this is not the deep learning of those organelles, just kind of naming them, right? But we have to learn them. We have to learn about them very deeply. It means one by one we need to study, right? So we'll do it. We are starting it from the plasma membrane onwards. We are continuing, right? Okay, but first of all of that, you have to understand what are the examples for single membrane bounded and double membrane bounded organelles, right? Okay, then we'll write the topic here for examples for single membrane bounded organelles. Single, we'll write single membrane bounded, single membrane bounded. Then here double membrane bounded, double membrane bounded, right? Single membrane bounded, then double membrane bounded, right? Okay, so first we'll go through the double membrane bounded because it is much easy, right? Okay, there are only few means first of all we'll go with the nucleus. Okay, nucleus. Then we have what? We have mitochondria. We have what? Chloroplast here. Mitochondria, nucleus, chloroplast. Okay, mitochondria, nucleus, and chloroplast. We have them as the double membrane bounded organelles. Double membrane bounded organelles. Okay, got it, right? Got it, right? So those are the double membrane bounded organelles. It means they have two coverings, right? They have two coverings. Okay. Then we come to single membrane bounded. We'll start from what? Start from Golgi apparatus. Golgi apparatus, right? Golgi apparatus. Then move to endoplasmic reticulum. It has actually two types. There are two types. One is RER. RER means rough endoplasmic reticulum, rough one. Then the other one is SER. It means the smooth endoplasmic reticulum, smooth one, right? Okay. Then after that, we have what? Peroxisomes. We have peroxisomes. We have what? Glyoxysomes. Glyoxysomes. Then we have what? Lyso. We have lysosomes. Okay. Then what else we have? What else we have? Okay. We have the vacuole. Vacuole. We have vacuole, a single membrane bounded organ. Then we have eukaryotic what? Flagella. You have to mention it specifically as eukaryotic, right? Because prokaryote also have flagella, but there are no membranes there, right? Then you have to write eukaryotic cilia, right? Prokaryotes also have cilia and they lack membranes, right? Eukaryotic cilia. Then peroxisome, lysosomes, ER, Golgi, vacuole, then eukaryotic flagella, eukaryotic cilia. Right. Yeah, those are the things we have to discuss, no? Right. Those are the things, right? So we can separate them like this. This is the separation, right? This is the separation. Okay, you can separate it. Okay, you can separate it like this. And one important thing here, right? Okay, one important thing actually, these all these both of these organelle types, they share the common structure found in plasma membrane. It means plasma membrane has a common structure, right? Plasma membrane has a common structure. Then what? Double membrane bounded organelles, their membrane also share that structure. Then single membrane bounded organelles also share their structure. It means structure of the plasma membrane can be seen in both single membrane bounded organelles and double membrane bounded organelles. Got it? Very simple fact. Okay. Now you can complete it. Huh? You can complete it. Right? So you have to figure out what are single membrane bounded and what are double membrane bounded. Only three for double membrane, right? Okay. So nucleus, chloroplast, and mitochondria. These are found in eukaryotes, right? Actually, these are found in eukaryotes, but the flagella cilia also found in prokaryotes too. That's why we specifically mention them as what? Eukaryotic flagella and eukaryotic cilia and eukaryotic flagella. Because actually prokaryotics lack organelles, right? They will have subcellular components. Got it? Right? Done? Completed. Ah, nice then. Okay, nice then. Right. Okay. We'll move to the next one. Then the topic is subcellular components, right? Subcellular component examples. We are writing them. Subcellular components. Subcellular components. Subcellular components, right? Okay. Subcellular components. So what are the examples for subcellular components? First of all, first one, they are the ribosome, right? Then cytoskeleton, then centriole, then centrosome. These are the examples for subcellular components actually. So these are what? These are not known as what? These are not known as what? Organelles, right? These are not known as organelles, right? Got it? These are non-membrane bound. These are non-membrane bound. Okay. These are what? Non-membrane bound. Okay. Got it? Non-membrane bound. Okay. Non-membrane bound. No organelles, right? Got it. Now one write it down. And you can write down the rest of the structures found in prokaryotes also, but we are mainly discuss about the animal cells, no? Therefore, we are not writing in any particular structure found. It means prokaryotic cilia, prokaryotic flagella, each and every kind of thing found inside the prokaryotic cell also comes under subcellular components, right? That's the simple theory there. Right. Okay. Then that's all, no? Subcellular compost. That is right. Okay. Now, most important fact. Now we are moving towards the important fact here. So now we are focusing on, right? Now we are focusing on the differences between the vast differences between animal cell and plant cell. Okay, major differences between animal cell and plant cell, and how we can differentiate them also, right? Majorly, right? Major differences. Okay, so then we can kind of have differences here. First of all, we will put plant cell here. We will put this side plant cell. Then here, what is the other one? The animal cell. Right. When it comes to plant cell, the outer covering is here, and it is known as what? Cell wall, right? This is known as what? This is known as cell wall, right? This is what? Cell wall, right? Animal cells lack the cell wall, and inside of the cell wall, immediately to the interior of the cell wall, we will discuss the structure of cell wall, all those things in a coming weeks, right? Here we just only focus on the name. Then inside that there is plasma membrane, and there is what? Animal cell also starts, it means begins from the plasma membrane, because there is no cell wall there in the animal cell. Okay. And majorly, if you go through a plant cell, you can see a kind of a large structure there. This is known as central vacuole. In animal cell, there is no such structure. And also when you go through the plant cell, you can see what? This structure called green color one. This is the called what? This is called the chloroplast. You can see the chloroplast here. No such structure in animal cell. And inside the cell, you can see what? The cytosol here. You can see the cytosol here, right? Cytosol here. That is common for both animal and plant cells, right? Here you can see the cytosol. And also to demonstrate these are living cells, we just rather kind of, we can see the nucleus here, and also when representing cells, living cells, we just put nucleus here, right? In order to show that this is living cell, right? Otherwise non-living cells, they lack the nucleus, right? Okay. Right, this is the arrangement of the plant and the animal cell, kind of a differentiation, major differentiations, right? There are so many differentiations. Other than this, you can notice there are some other facts also common, which are unique to animal cells, which are not seen in plant cells also. There are such kind of facts also there, right? Such kind of things are also there. So you have to keep in mind, right? You need to be mindful of that, right? Okay. Then, okay, right? Then uh, this is what happening. Then this is known as cell wall. That is common. That is only unique to. It is only found in plant cell. In the comparison, right? You can find cell wall in protista also, right? But we are comparing both animal and plant cells, right? Then you can see the chloroplast, which is only seen in plant cells. Chloroplast, okay. Chloroplast only seen in plant cells. Then you can see here nucleus. You can find it common to both the animal and plant cells. Nucleus, you can find it here common. Then also common factor, what is that? Plasma membrane. You can also find plasma membrane in these both cells types, right? Okay, plasma membrane in animal cell and as well as in plant cell. And also you can find the cytosol in both of these cells cytosol inside. You can see there, right? You can find it. Okay, so this is the rough differentiation of animal and plant cell. So major key highlights, it means once you first go through a cell, first view under a microscope, what you really notice, first of all, in a plant cell you can differentiate it from an animal cell with the presence of a central vacuole. You have to name that one also. This is central large vacuole. This is central large vacuole. You will notice a central large vacuole, and you will notice outer covering cell wall, and you will notice green color chloroplast. Got it? Right. So when comparing with the animal cell, they lack those three structures. Okay, then you can find out this is an animal cell. This is the plant cell. Note this down quickly. Quickly note this down. Right. So those are the key differences we can notice. Those are the key differences we can notice, right? Okay. Then I think you have finished drawing. Okay. And there's another very important fact when related to cell. So these are very important in future lessons, right? Okay, future lessons. These are very important. Once you just realize these terms there, then you will note down. You will remember. We have done that. All right. Likewise, you will understand it, right? Okay. So now then, when moving for the next one, so we'll discuss that one there, right? Okay. So this is a very interesting fact here, interesting one. That is, right, you can see now, you get to know with the plant cell now, right? So this is actually what, this is the plant cell. You will note this down because we have now just now we have discussed this is the plant cell because this is a cell wall outer covering, and you can see the what, you can see the plasma membrane just immediate to the cell wall here, and you can see the central vacuole here. Rest of that central vacuole will note it down, right? Okay, central vacuole here, you can see the central vacuole here, and you can see the what, you can see the chloroplast here, right? This is the plant cell, and you can see the cytosol. Cytosol where the organelles are embedded, right? Cytosol is the place where organelles are embedded, which is common to all living cells, right? You know that one, right? Okay, it is a kind of okay, semi-solid jelly-like structure. Semi-solid jelly-like structure. We have discussed that those, right? Then this is called the what? This is called the plant cell. This is called the plant cell. This is called the plant cell. Then imagine we are removing what? We are removing the cell wall out of that one. We are removing the cell wall out of that one. Then you can see, you can still see this central vacuole here, and you can still see the what? You can still see the chloroplast here. I just missed one. What is that? Is the, sorry, that is the nucleus. Nucleus, we missed that one. And here you can see the nucleus here like this. You can see the nucleus. So here you will note down this one, right? The difference is there is no cell wall as when compared with the early stage. There is no cell wall. What you can say to this? A cell without a cell wall actually is what? A plant, sorry, an animal cell. Am I correct? Can you write it as animal cell? Why? Why you can't? Because this is actually not the animal cell. It's not the only factor. Because you note down chloroplast here. You can see, you can see what? The central vacuole there. Are there central vacuole and are there chloroplast in animal cells? No, no, never. So then you can't write this, or you can give a name for this as the animal cell. You can't give a name here. You can't give a name, right? Okay. So actually this is a kind of a special structure kind of thing, right? We name it as, right, protoplast. Got it? Mind you, protoplast, right? Very important factor. Protoplast, right? Protoplast. Then you can see a relationship here. What? Plant cell. Got it? Plant cell minus cell wall equals what? Ah, nice. Protoplast. This is a very important point. Plant cell minus cell wall gives out protoplast. This will be very valuable when you come to lesson number 4. There's a part called water potential. There's a lesson called water potential. When you come to unit number 4, this fact, this relationship is very important. When the cell wall is removed from a plant cell, it gives out a protoplast. Okay, a protoplast. It is not the animal cell because it also has central vacuole and the chloroplast as well as they are in the plant cell. The difference, only difference is still this is kind of a plant cell origin there. Only difference is there is no cell wall, no this down. Okay, so plant cell without a cell wall is known as a protoplast. Got it? Very important fact here, right? right? Then there's another important factor, right? Okay, there's another important factor, right? So you need to note this down. Now, now, did you kind of think whether how these membranes are derived from? Didn't you have such kind of a problem? How these membranes are derived from? Okay. Now you can see this is the plasma membrane, right? Mind you, this is the plasma membrane, outer covering of the cytosol, plasma membrane, outermost in animal cell and in plant cell, just inside the, just interior of the cell wall. This is a plasma membrane. Then you might see, right? You will notice what? You will notice organelles like this, right? Double membrane bounded. Okay. Then you will notice double membrane bounded organelles like this. Got it? Double membrane bounded organelles like this. Then you will note down, you will note down single membrane bounded organelles like this. Then you will note down single membrane bounded organelles like this. Then you will note down single membrane bounded organelles like this. Then you will note down, right, double membrane bounded organelles like this. Then you might note down single membrane bounded organelles like this, like this, several kind of organelles, right? You will note down. Now here, see, plasma membrane has a unique structure, right? We'll discuss it next day. The first topic of the next day is the structure of plasma membrane. It has a unique structure. Keep it in mind. It has a unique structure. It has a unique structure. Okay, plasma membrane has a unique structure. So that structure is shared with all the other membranes. It means all the other organelles, they have that plasma membrane structure. If you take nucleus, it has. If you take chloroplast, it has. If you take mitochondria, it has. If you take Golgi, it has. If you take ER, it has. If you take peroxisomes, it has. If you take lysosomes, it has. If you take glycosomes, it has. If you take eukaryotic flagella and cilia, it has. They all have the same structure that plasma membrane possesses. It means the structure of the plasma membrane is shared with rest of the organelles. What? It is shared with rest of the organelles. Got it? It is shared with rest of the organelles. So all the single membrane bounded, all the double membrane bounded organelles, they all have the similar structure, right? Similar structure. It means the structure, if you take the structure of plasma membrane here like this, if you just kind of take a certain part, so this structure is found here, and structure is found in this one, and its structure is found in where? This one, and its structure is found in what? This one. Its structure is found in here, and its structure is found in this one, and its structure is found here, and its structure is found in this one. So therefore, each and every organelle possesses the same structure which was there and which is there in the plasma membrane. Got it? So if you take the structure here, if you name the structure, the composition here, if you understand the composition here as X, then the composition here is also what? It is also X. Here also there can be slight differences, right? Mind you, there can be slight differences, but majority of the time, more than 95%, they share the common structure. There might be some different modifications according to the places they are in, according to the function they are possessing. There might be some kind of modifications, right? Okay, there can be slight modification. They are not very major modification. They are very minor, right? Sometimes they are negligible. Okay, got it, right? So that's why you name all those organelles share the common structure of plasma membrane. Note this down quickly. Note this down. Note this down quickly. Okay, then write down this one. All the single membrane as points, right? Write down. All the single membrane bounded organelles, all the single membrane bounded organelles, all the single membrane bounded organelles, and double membrane bounded organelles, and double membrane bounded organelles shares shares the common structure, shares a common structure. Both plasma membrane, then single membrane bounded organelles and double membrane bounded organelles shares a common structure. Shares a common structure. Okay. The plasma membrane, single membrane bounded organelles and double membrane bounded organelles shares a common structure. It means that the structure of the plasma membrane is similar to the structure. It means the membrane structure of single membrane bounded organelles and double membrane bounded organelles are same, ne? Right? So within brackets write down. Within brackets write down. Within brackets write down. Their structures are same. Their membrane structures are same. Their membrane structures are same. Right. Then write down prokaryotes lack membrane bounded organelles. Prokaryotes lack prokaryotes lack membrane bounded organelles. Prokaryotes lack membrane bounded organelles. Okay, right. Then write down eukaryotes 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 possess more efficiency possess more efficiency possess more efficiency eukaryotes possess more efficiency and more order and organization and more order and organization and more order and organization as each and every as their each and every as their each and every as their each and every organelle 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 is covered by organelle is covered by membrane covered by membrane. Is covered by membrane, right? So actually, because of that separation, it means they are separated now. They are found actually kind of separated now. Imagine in prokaryotes, all are found there without any separation, because no membranes means no separation. But eukaryotes, they have separated. It means because of the membrane, organelles are separate from each other. You can see they are separate from each other. If there are no any organelles, then you can see all of things are there as a bunch. So that decrease the efficiency and the order, right? That's what happened in prokaryotes. Okay, got it. So those are the very important aspects of the arrangement of organs. It means arrangement of subcellular, it means cellular components, they are which are enclosed within membranes. That's the use of they are being enclosed within membranes, right, when compared with what subcellular components, because they are non-membrane bounded. They are not they are not embedded, or they are not enclosed within membranes. Got it, right? Okay, that's it, what we have to discuss, right? Okay, then we'll have a quick kind of revise here. So first of all, we discuss about cellular components. It means the components which are within the cell, found within the cell. We can divide it into two as organelles and subcellular components. Then subcellular components, they are the non-membrane bounded. Then organelles, we can further divide them as single membrane bounded and double membrane bounded, mean with membranes. These are without membranes, right? Then we get to know about the examples related to single membrane bounded and then double membrane bounded. We got to know about the examples. And here, very important fact that when you mention the flagella and cilia, you have to mention it as eukaryotic, because prokaryotes do have flagella and cilia, and that those are non-membrane bounded. That's the main reason. Okay, there are only three double membrane bounded organs. We just commonly we have to discuss: nucleus, mitochondria, and chloroplast. Then plasma membrane structure, we can see it in both single membrane and double membrane bounded organelles. Then subcellular components, they are actually not considered as organelles. Ribosomes, cytoskeleton, centriole, and centrosomes are the examples. Then when you quickly go through two separate cells, if you just go through the microscope and see the kind of highlighted things, major aspects of the differences between those animal cells and plant cells, what you will note down, in a plant cell you will see a central large vacuole, not in animal cell, and you can see the chloroplast there, and you can also see you can also observe the cell wall there, and all those things are lack in the animal cell. Then when come to plant cell, if you peel off the plant cell wall from the plant cell, then then that structure formed afterwards is known as protoplast. It means kind of a cell. Actually, it's a cell without plant cell without a cell wall. So plant cell minus cell wall gives out the protoplast. And finally, we got to know that the common structure, it means the common structure found in plasma membrane is shared within rest of the organelles too. Right, the common structure found in plasma membrane is shared within rest of the organelles too. Got it? Okay, it means they all have the same structure, right? That's it. So that's it what we have to okay, that's it what we have to discuss for the today's proceedings. Hope you guys have understood the better of today's explanation. So just compare with comparison is not actually it's not a good thing, but comparison in your notes is a good thing, because you can compare, you can get an idea of the quality of the class, right? That's why, right? So you can compare with your friends with their notes. You can see whether these facts are given or taught in their classes, right? You can understand it, right? So then that's the important we are carrying. Actually, these experience I have gained those experience from teaching revision students, because they lack those parts. So therefore, for you, I am teaching those facts, because then you will have a perfect class with full theory, full application, and full these important facts, right? Okay, got it. So study those facts, very important. Just go through the note once again and study. Then we'll meet on next week. And also, as I explained, as I told you earlier, there will be a special grand seminar coming soon. So just mention you or share it with your friends also. We'll publish it, right, as quickly as we are in the dates and times. Okay, got it. So till then, have a nice day. Okay, good night, and we'll meet on next week with the plasma membrane with the topic. Okay, then have a nice day. Goodbye.